So yeah, thanks you for the kind introduction, and I'm grateful for your presence today. So without further delay, I would start. Along with the cancer and cardiovascular diseases, one of the most common reasons for the deaths around the globe is medical error. So there have been extensive literature published, in last couple decades. It's around like over 200,000 available through PubMed, which discusses in different ways, the misdiagnosis or delayed treatments. So one of the trending subjects, towards this cause is precision medicine. And talking about precision medicine, this initiative has been taken by, former president of United States in 2015, and that's what the focus of my lab is. Talking about precision medicine is mainly providing, personalized treatment to the patients by looking at the different aspects of the data coming from the patient, which includes this lifestyle, genomics, phenotypic, metabolomic, and etc. As we all know, whenever a patient goes to a clinic, different kinds of diagnoses have been conducted, and if required, different kind of procedures have been performed. However, the question comes that one treatment or one treatment for one patient, or one medication for a patient might be good, and another patient with a similar symptoms with another race and age and gender, that diagnosis and medication might not be good as well for the same patient. So, so to investigate this cause further, we look into the genomic profile of the patient. It's been over 100 years since we are studying genes, and there have been enormous amount of data which have been generated. However, the challenge around is about the data interpretation. This data has been generated with the collective and effort of different components sitting in one institution, or variable components of variable institutions, like starting from healthcare institution like in US. We can send patients in clinics, and then research investigators start different projects, and core and bench support data generation, and computational scientists using cloud computing and high performance computing environment do different analysis. The goal of my lab is to do some translational research so we can take those results back into the clinic. At every stage of this institution, different kinds of data is generated. Looking, if we are living in an ideal situation, we might expect that these data have been very well integrated and very well shared and analyzed, and translated from research community to clinicians. However, in reality, this has not been the similar case. It's very chaos. It's the data structures, high volumes, it's completely heterogeneous, and it's not easily possible to communicate data on time. Sometimes patient is dead and then the results are coming. It can get a publication and a grant, but not the life is saved. Yeah. So that's what, so in this situation, how you gonna, support the precision medicine? So the question comes like, how you gonna, we are not here, especially my lab is not here or focused to develop new technologies, but how can even we use existing ones to support the cause of the life, right? So this is what we are focused on. My lab at Rutgers, we develop technologies. We are good in developing infrastructures to support PHI data. We are developing different applications to pull data right away from the Epic and health systems from the clinics. We are good in developing different tools and technologies for orthodox bioinformatics analysis. And then we are looking and implementing machine learning approaches. But this talk is not about all of these things. But only I'm focusing on one of the tools or applications we have published and presented recently, and that's called GVViZ. This application is mainly, proposed and developed to support gene expression and gene disease annotation analysis within the clinical environment for the support of translational research. So, yeah, so this is a very simple tool. I will not waste your time in discussing in detail, but I will give you a small tour of it and one case study as well. So first of all, it starts with the connection to the databases. Second most important part is when RNA-seq data have been generated of the patients, we have developed a pipeline which is not a secret anymore. Like everybody is using a similar kind of pipeline. However, the results and the QC matrices produced by this pipeline have been automatically, saved in a centralized database server which is connected to the GVIS. Then the next step is gene disease annotation. So the question is like, for example, if you are in this room, there might be, we are a community of researchers. Somebody will be working on cardiovascular disease, somebody is working on cancer, somebody is working on diabetes, and etc. and etc. So how can you all use one tool for your research? So if you want to look for the genes, like I am doing heart failure based research. So if I am interested in looking at all the genes available, which are associated with anyways with the heart failure or further diseases, how can I do that? Like So for that, first of all, we started looking into the database, Google, the first search engine, then finding different databases and which database to start with. So at that time, we spent almost over a year to collect the data from several data archives and combined into a unique database where we have already published in briefings and bioinformatics over research on that. And then we have, produced a database of over 100,000 gene disease combinations and over 1 million gene SNP and disease combinations to support EHR system. And that is also available through the apps and also published application. Next, we recently published last month another database, which is based on, solely on genes approved by the ACMG college. And we have connected those genes to the ICD codes and then back to the healthcare records. So this database is collectively is a backend of our GVViZ tool where you can talk and communicate regarding diseases for the patients. Last but not the least is a visualization part. You can produce different kind of visualization, heat map. This is not, I will not say we have a fancy visualization, but we have an effective visualization. And then you can export the results for further analysis, right? So these are some examples you can see here. We have some Alzheimer-based example, arthritis, diabetes, and it can support you for over 6,000 different kinds of cancer disorders as well. So going forward, this tool is publicly available. It's open source. There's a demo available, as you can see at the end of my slide as well. And this paper is published over a year ago. So then the disease which is close to my heart is the disease we explore further in our lab, and that's the heart diseases. So, I will give you a small use case of our tool that how we have efficiently used to study heart failure, atrial fibrillation, and some other cardiovascular diseases. So we have recently designed a study, and where we collected the data from EHR, and then we consent in some patients, and then we draw the blood sample, extracted RNA, sequence it, and then did a gene annotation and gene disease annotation analysis and expression analysis using our tool. And then one another good feature of this, like we all are going towards artificial intelligence and machine learning, the outcome of GVViZ can also be used for that kind of analysis. And there's an example of it in the later part of it. How much time I have? I'm good? Okay, yeah, good. So starting from this is a small example of 61 patients cohort. We have a tool built for efficiently extracting data from the health system, which is also published in open source. And these are some examples. I hope it's working. Yeah. So in the top part, we have an example for heart failure. On the left side, you can see it is telling you about the diseases. On the right side, it is connecting with the genes. And on the bottom, it's the number of patients and the number of control samples. There you can see the difference in the pattern between, if I say versus, disease and versus healthy patients. Same goes for atrial fibrillation, and same goes for some other cardiovascular diseases. So it exports you a gender classification, race-based classification, and we have also validated our results using orthodox gene expression analysis, gene enrichment pathway analysis, which is part of the paper we have published recently. Then what we did, this cohort which I just showed you, we have also produced the whole genome sequence data. My lab is actually focused on producing 5 different kinds of data of same patient. We are doing our whole genome sequence, RNA sequence, and and behavioral, metabolomics, and clinical data. So I'm wrapping it up. So these are some findings we have. If you're interested, these are the 3 papers based on the outcomes produced by the GVViZ we have recently published within the last 6 months. Then the last part of it that we have recently, published an article in briefings that we have, looked at all the applications and approaches available from machine learning for gene expression data, especially. We produced our new pipeline. We are also focused on producing portable AI/ML pipelines. Like you have other applications you can download and run in your local computer. One of the examples is already there. We applied the same pipeline to our data produced using GVViZ, and we have published paper in April this year. So that's also there. If you are interested, the time is less. I won't focus on that much. However, we have some interesting findings. For example, we have high accuracy. We have used random forests for this one, and then we observed age and gender appear to be high correlation in HF and race versus for the AF. So I would like to summarize my talk. We have some, introduction to the GVViZ today. Going forward, we are linking AI and integrating variance analysis in that. This is my lab. We are 3 years old at Rutgers, and these were the students working with me for last 3 years, and this is my current lab. And I have some funding received from NIH and some different other resources, and I have some strong collaborators working with me, including MIT and Broad. And I thank you very much for your attendance, and if you have any questions. Yeah.
Thank you. Any quick question for Zeeshan? Hi. So, what's the advantage of connecting the ICD codes to the ACMG pathogenicity criteria? Because presumably a pathogenic mutation is going to be pathogenic across diseases.
It's a very good question. Thank you very much. Actually, if we want to take the results of our science back to EHR doesn't work on like if you say to the clinician diabetes, then he will ask what diabetes. There will be 500 codes for it. Which code you are talking about? That's why we need to connect ICD code with a disease and disease with a gene to make it specialized and compatible to the EHR systems, electronic healthcare record systems.
Thank you.